now we have explored a little bit on the differential gene expression and also in the gene set enrichment analysis. So you know how to get the functional information out of your results. But in that case, we have quite simple experimental design, just two groups and the simple comparison between them. But that is very uncommon to find in RNA-seq analysis. So we work with very complex experiments and models, and we want to put everything into perspective. So the differential gene expression is based on what is called the generalized linear models that Roy mentioned, and I'm just going briefly over it and explain from the basics for you to understand how to model complex data or complex experimental designs. So GLMs, they are generalization of the linear regression analysis that you probably know, I assume. But rather than modeling that with the normal distribution or as a result, we can have any kind of distribution associated to it. So we can use inverse Gaussian, Poisson, which is you probably know for microarray, or binomial or negative binomial distributions. And I'll mention a little bit about those distributions right now. So a normal distribution, you're probably familiar with that. So for a log normal distribution is when we transform the counts to log. So this is basically the kind of distribution we have. A negative binomial distribution is the raw counts that we get from the RNA-seq, and this is where DA-seq2 and HR usually are based on. So they improve these negative binomial GLMs. So negative binomial is based on count, so 1, 2, 3, and that's why it is a spike like this rather than have like a curve, because you don't have values in between. The Poisson will be very similar to the negative binomial. And I had a question before, what is the difference between those two? And that's one of the reasons why DSEQ and HR actually excel at differential gene expression with low amount of samples. So if you compare a Poisson and a negative binomial distribution, if you have very small samples, so this is a parameter that you can assign to the distribution. So either you have low amount of samples, small sample size, or low counts, genes with very low counts, so they have higher variance. So this value here will be small in the distribution. And then you can see that the Poisson and the negative binomial distribution are quite different, because the negative binomial will then accommodate for these shifts in what is called the overdispersion of the different genes. On the other hand, if you have genes with very high amount of counts, or if you have a very large sample size, so many replicates, then the negative binomial distribution will be very close to Poisson. In fact, the negative binomial will be basically converged to a Poisson distribution for a very large number of samples. And that's why when you consider that either if you have low amount of counts, you have a more variability, that variability is already accounted in the model when you do differential gene expression. So that is one of the reasons why we use negative binomial in HR and DSEQ2, especially for RNA-seq data. But let's now get some intuition about how GLMs work. So what if I have two groups, just like we had now, control and drug A, and that you probably saw in the results as well. So then you can assign to the metadata as a factor, as Roy mentioned, and you can control which level is that you're comparing to. So this control and then drug A, but you can see that this one will be called what is named an intercept. So everything, all the other groups will be compared to the first group that you define in your factor. And that's what Roy was mentioning in his lecture. So this should be always the first level of the factor. So the group that we're comparing to, or like the baseline. And then you have control and drug A. In your table, you probably will see that you wrote a formula similar to this. So your gene expression is proportional to the drug, and the drug is this factor that you have in the metadata. And then for this gene, interferon gamma, that I'm explaining here, drug A has an effect size of 1. Here I'm showing FC, but you can mention anything depending on which test you're using. In EDGAR or DSEC, that is log fo change, which is written in the top here. But so the idea is that there is only one column. You don't see a column for the control here, because this is already been controlled for this expression here. And that is the distance that you can see between the control and the drug A treated group. But in some cases, sometimes also you can, you want to test if the control is actually different than not having anything. And then what you can do is add a 0 here. And now the 0 will be the intercept, this intercept here. So we're comparing both control and drug to 0. So, but here, please note that we are testing if the base expression is also different than 0. And then you probably will get a significant p-value because, of course, we have a value here. If you have counts that are not 0 or that are all 0 for one group, then that would be false, because then all counts are 0. Is 0 different than 0? No. Yeah, so in general, what we want to do is compare between groups, and then this is what a formula that you should write. So this is what we saw already in the exercises this morning. Do you have any questions about this? No? So what if we have now 3 groups, drug A and B? So then in the metadata for the drug, you can also define that and specifically say control drug A and drug B, if that is not sorted automatically for you. Control will be exactly, just because I put them in the beginning, that will be the intercept or the first factor. And if I do Y over the drug, then I'll have the gene list, drug A and B, and the effect of those, and then a p-value. And the drug A, you can see it's a increase in 1, and then drug B, an increase in 2 in the gene expression. And remember now that you can have these results, and then this p-value now means that we're testing if that gene is significantly different than 0 in any of those conditions listed. So now we are having a p-value with all the tests that we're performing, with all the groups. And that's basically how you're gonna do. If you have 4 or more groups, that is exactly the same result that you're getting. A table with the results for every group. And by default, if you have the drug A and B is 1 and 2, the control is always 0, because we're comparing to that one. But what if we have 2 variables? So like now we have trying to see the interaction between them. And let's say that we have drug A and B, and then you have another condition that you have both drugs added to your experiment. Let's say either a plate or a group of mice treated with both drugs. So here you can see that A has an effect of 1 if you just add A, B has an effect of 1. But if you sum of the drugs together, you don't have an effect of 2, which is the effect of A plus the effect of B. And this will be the expected effect. But rather, you have an additional one here. You have an effect of 4. So that means that drug A and B have an interaction factor that is to add 1 to this experiment design. So what you should avoid doing is, whenever possible, is not doing this to have the control drug A, B, and drug A plus B, if you want to find that what is the interaction between the drugs. Because now what you'll have is that drug A is equal to 1, drug B is equal to 1, and drug A and B is equal to 3. That the effect of the interaction is equal to 3. And that is not true, because I'll show you in the next one. And then you have a very significant p-value in here. What you should do instead is then define drug A as one metadata, drug B as another metadata, having the control and drug A, control and drug B. And then you write a formula, Y is equal to drug A plus drug B. But in this case, it's also not enough, because now we're averaging this group, the blue with the purple and the red with the purple. So then you have the average of all those groups. So just adding is not enough. For this one, what we want to do is that have what is the effect of drug A, what is the effect of drug B, plus what is the interaction effect between those two drugs. And that can be rewritten as the interaction and all possible combinations between drug A and B. It's with this asterisk sign. If you have many more drugs, you just put another asterisk and drug C, D, and so forth, so on and so forth. So now in your table, you'll have drug A, drug B, and what is the interaction effect between them. So now if drug A and B have the effect of 1, the interaction effect here is exactly what were expected, is an additional 1 in signaling response for this, this gene. And this is the corrected or the correct interaction effect that you would expect. Any questions on this part right now? Or is pretty clear? Yes, I don't see everyone, but I'll assume that's a yes for now. Yeah, so this will be the most correct way of doing your analysis. But now, what if I have a batch effect? Because now I have a factor that I can actually model in my data, but I don't want to have results based on that. Because in the previous one, if I use the p-value that I'm getting from here, I'm getting still, even if the batch effect is a difference in my data set. But now let's say that you have drug A and 2 batches, A and B, and you can see that the batch 2 is a bit higher than the batch 1 by 1, but it's still the drug effect is still 1 in both batches. So that can be modeled using the addition here. So the gene expression is batch plus the drug A. And you saw also in the differential gene expression, when you want to list your results, you select a coefficient for a result, or at least you have the option to do that in both the ELIXIR and HR. And that is showing, for example, like now show what is the effect of the batch and the drug, just like as in the previous ones. So the batch has effect of 1, the drug has an effect of 1, and that is very significant. But if I'm not interested in the batch, I just want to model the batch, but not use that for knowing the effect of the drug, because that's my question, what drug A does in my experiment. So when you model, you should model both of them together, and then just look at the results from the coefficient 2, which is the drug A. So then you will get a table with what is the effect of the drug A and the p-values. And here you can notice that the p-values will always be different in this case, because now you're testing different things. The first one, you're testing, does the batch or the drug A impact on my analysis? And here is very yes. But then, of course, here now I'm testing, does the drug A impact on my results? And yes, and then you have a different p-value for that. And that is because you're now selecting only one coefficient to test. So this is the right way how to do your analysis, including batch effects as well. So you don't need to clean your data in a way, but you can just add this to the model, and then the list of significant genes will also take the batch into consideration. Now let's get to the more complex experiments. Now I have 5 patients and I have 2 visits, as you can see here. So every patient here is now assigned where it is in the other visits as well. And I have 2 drugs that the drug A actually is just one drug that was treated or not in the first visit or the second visit. Actually, these ones should not be linked actually. But then I want to know what is the effect of the patient, what is the effect of the visit, and what is the effect of the drug itself. So then if you type this for all coefficients, this is actually not just 1 and 2. No. This is for all of them. Go back. So here you'll find what is the effect of patient 2, 3, 4, and 5. So it's like this difference in height between the patients. What is the effect of the visit, which could be considered as a batch effect? Maybe I don't care exactly when they come, but it's probably just a problem in the data collection. And what is the effect of the drug A across all of the groups that we have here? And then you have also a very significant p-value. But what you should do then is, of course, model all of them all together, but then just take the coefficient 6, which is what is the effect of a drug, considering that we have different patients and that we have different visits or different batches. And in this case, also the p-value will be different. It doesn't need to be small or bigger than the other one. I just want to emphasize that it will be different testing because you're testing different hypotheses.
I have two questions here.
Yes.
The first question is this coef equals 6. Is it a parameter of the function of the GLM function?
Yeah, so when you do, I think it's in the WALD test for the DSEC2. I need to double check later the code. But then you select, when you export your results table, you do coef equals 6. It's inside the function there.
Yeah, okay.
Yeah.
Good. And the second question is, when we use this coef equals 6, can I understand it like this, that we tell the program that it should normalize in a way over the patient and the visit so that we have the true result of the drug?
So the coefficient is basically running the test only for that variable. What happens actually is that this table will be generated if you run this model. It will be saved in the DSEC object. And here, by default, will be calculated the p-value considering the whole table. But now if you do coefficient 6, I will just take this value. This one will not be changed. Is like what is the really effect of the drug. But then the p-values will be calculated based on this one, only on this one. Does that make sense?
Yeah, okay.
Yeah.
But
It's
so
just
if you look at if you look at the complete table, then you see all the p-values, right, for each effect. Like if it's a normal GLM, that's what you get.
Yeah, no, exactly. If it's a normal GLM, you do that for every factor. Yes. And plus 1 for the whole model. And then here in the DSEC2, this is how they usually display. And then you do the p-values afterwards when you do this coefficient to know for every factor. But in essence, it's the same thing. So rather than having like 1 GLM per gene and then you have a rows, each factor listed by rows, DSEC just transformed it like this to have what is the effect of every variable. And then the testing is done separately using these coefficients. But it essentially is the same thing, yes. However, I mean, here is just a simple example. Let's say now that, for example, the patient and the visit maybe has interacting terms. That might even be the case. And then you need to put an asterisk here. So all those formulas are not like a rule of thumb that you need to use that one, but it's just an indication how you can handle that kind of data. It might be that patients will vary depending on the visit or for some reason, or maybe the drug and the visit actually will have some interaction term, and that can also be modeled here just by changing the plus sign with an asterisk. And that's up to you to explore because that's really dataset dependent. But in general terms, for batch effects and for patient level information, for the second HR, then you do exactly as I'm showing here now. So adding those effects. What if I now have a time series? And I saw that many of you already have those kind of data. So now it's important then to set this time as a factor, unless you want to really know that the time and the gene expression follows let's say a correlation, or will follow kind of this trend. So what I can do is then, if you have, for example, sample groups, which is this case here, rather than scatter points, then you can add a string And again, set them the day 0, for example, or the beginning as your intercept, as in the previous ones, instead of having 0, 1, 2, 3, or 0 to 4 and 6. And in that case, you will have a table with these expression values. So day 1 is increasing in 2 and in 1, and the other days in 2, 1 and half. But the problem is that sometimes there are other variables, for example, patient age, exposure time, or if you have very complex data sets, those measurements are not making a group, because you're not grouping the patients based on the age, but you want to consider the patient age in your differential expression. Then you should leave those as numeric. If you have the patient age or some kind of exposure time. And there are many other variables that also follow this trend. But in those cases, you should leave them as numeric. Because if you leave as numeric, what you're measuring is not the group information, but rather the slope of the correlation curve between your measurement, let's say interferon gamma, and the time, or the patient age, or the exposure time, and so on. So this is what you will get. You'll get just one column, and this number here will be the increase, or the slope is exactly the slope. So increase of 1 of interferon gamma per 1 day in this example. So if you have 1 is here, and have 2 is 1 basically here. So it's 1.5 minus half. So this is the slope that this is showing here. And that is good if you have patient age or other continuous variables. But now, what if I have the time series and a treatment? And I saw that many of you also have that kind of interaction. So then it will be exactly the same thing, time times the treatment, which is exactly the same as the time, the treatment, and the interaction between them. So overall differential gene expression at any time point, the gene expression between the conditions, treated or not treated, and the differential gene expression per time point, specifically to that time point. And for this example here, you have the gray and the red ones, and you see that this red one is a shift of 1 above here, so it's a little bit more specific to this one. This one is shifted a little 1 down in this example here for day 4, for time 4. So what is the effect of 1, 2, 3, and 4 is adding 1, 2, and then 1 here, and then half. What is the effect of the drug in general terms of adding 2 to every time course? And what is the effect of each point in the related to that drug? So the 2 first time points is not adding anything. The second time point is adding 1 additional, so it was supposed to be here. At time point 3 is not happening anything, and then at time point 4 is -1. So it's, and this is very significant. But again, what is your question when you have these kind of very complex data sets? So here we're testing if any gene is significant at any time point at any interaction. And that is really good if you have a heat map, for example. Then you can cluster your gene expression patterns and look at them individually. Or maybe actually you're not interested on the specific time points, it's just for what is the effect of the drug. Then you can just use this one for the coefficient and test it specifically, just like as in the previous ones. Or you can test what is the effect over the time points or specific effects over the time points. So now this will really be specific to your project. What is your question in there? And actually all of them, all of those results are important if you're in the paper. So you describe what is the effect of the drug. These are the genes, and you show an enrichment. What is the genes that change over time? These are the clusters, these are the genes. And then which are specific maybe to a time point. Then you pick out which ones are important for you. And I just want to have a reminder on the meaning of p-values. So one of the things is we did after differential gene expression and also in the gene set enrichment analysis is that we correct the p-values by FDR, for example. One of the reasons why we do that is because by chance, at least 5% of what we consider significant are probably not significant. And those are considered the false positives. So imagine that you have the p-values of all the tests that we did. So we have 10,000 genes. We test between the two groups if they are significant or not. If there is actually no difference whatsoever between our groups, the p-value distribution should follow this blue part here. So all of them will be very well distributed, including some genes here in the beginning. For example, 0.05 is about here. And you see that there are some genes that are significant because they are significant to us in this concept of being less than 0.05, just by random chance, even though there's no difference between them. If there is difference between them, the p-value distribution should be something like this, that has a spike specifically at p0005. And you saw actually that in the differential gene expression lab, that you see one bar high and all the others very constant. And that means that there is some difference, and that difference is very specific. There are many other variations of this diagram, and they can change a lot depending on how many samples you add, which groups you have, do you have batch effects, and so forth, so on and so forth. So here, for example, you have about 6,000 differentially expressed genes. This one is also a little bit that. Here, we would assume that this one is even though this bar has 2,000 genes in here, most of those are probably not differentially expressed actually. And this one also has 5,000. So always take p-values as with a grain of salt, what you consider significant or not. And that's why we do FDR correction when you do multiple testing to adjust for those that are 5% to not become significant if you have a null hypothesis, which is this one. But the idea is to reduce false positives, but you will not eliminate them in any way. And another thing that you should remind is that the p-values represent the confidence that you have in your measurement comparing the groups. So for example, and not showing that the groups are actually different. So for example, if you model the drug comparing these two conditions, control and drug A, with an n equals 5, and then you see p-value 0.1. Oh, it's not significant. But let's just add 20 samples instead. Now we start getting a little bit significant, and that's what UPI usually say. Oh, no, let's do the experiment and add more samples. But now if you add 200 samples, now it's very significant. Oh, good. But what actually the p-value is telling you here is that how accurate or how sure I am that this 0.2 is actually 0.2. P-values indicate how accurate I am in measuring this effect size here. So 0.1, that means I'm not very accurately measuring 0.2 because there are some sample variations. If I add more samples, I'm estimating this difference 0.2 more confidently. And that's why we always need to check the effect size or the log fold changes in the RNA-seq. When you have this volcano plot, we saw the fold changes and the p-value, and we set cutoffs in both sides. And that's because of this effect. So the FDR, and just remember that the FDR does not correct for this one. So just by increasing the amount of samples you have for comparison, you will make your results more significant. And that's not only valid for RNA-seq, that's valid for any test that you do, even including your t-tests. This is just one example of that. So a comparison of genes in microarray and RNA sequencing. And then here you have 100 samples or 200 samples. And here I'm even having a very strict cutoff, FDR. We have hundreds of samples. And you can see that the p-values increase quite a lot if you have twice the amount of samples. But you can see that the fold changes do not change. They are basically flat. So the fold changes are not impacted by these differences in sample sizes, but the p-values are. So keep that in mind when you think, oh, nothing is differentially expressed, or maybe you have, oh, I have 10,000 genes differentially expressed, but 10,000 is basically half of the all annotated genes, and that's very unlikely. So you need to have these analyze this with some biological information. And that is it for today. Not for today, sorry. We're not finishing now. For this consideration. Do you have any questions now that you know maybe kind of which test you should do or how you should analyze your own data?